slide, okay, you can actually see here that there is a general question posted here. Okay, meron tayong general question like, is this water safe to drink? Okay, this is a very general idea. So this is a general question. The problem in a certain community, for example, is their water. Okay, so when they were able to get water from a balloon or perhaps or to a well, so merong complications like diarrhea and other um, diseases that they were able to observe. So with that, they were able to come up with this problem. So meron silang, for example, um, is the water safe to drink on that well or in that balloon? So if we were able to identify this kind of problem or general question, we're going to translate it. Ano ba ang paano ba natin i-translate itong mga general questions? We have to go back to the idea of analyzing a certain sample. So dito sa ating specific question, what is the concentration of arsenic in the water sample? Or it can be, what are the different organisms present in the water? Pero yun kasi very biological. Very biological and pwede ring microbial in nature. Okay? But here, we are what do you call this? Determining the constituents that are present in the water sample. So dito, sa binigay na example is arsenic. Arsenic is a very toxic element when consumed. So it can result to even death, severe case. Okay? Ayan. So yun yung pinakauna nating process. So we have to formulate the question, identify the problem, and translate the general question into a specific question. Okay? The next thing that you're going to do is we have to select analytical procedures. So in selecting analytical procedures, there are certain criteria we need to follow. So first, ang mga nakalagay dito ay yung mga limit, uncertainty in measurement, and even limit of detection. So we have to determine the limitations of the procedures, and even how does the procedure affect the sample perhaps, no? Kaya sabi dito, destroy sample. So it may confer or destroy the sample perhaps. So we have to look on those cases or criteria. And then yung lagi kong sinasabi sa inyo, yung availability ng resources, availability ng equipment, ng chemical reagents, and even the time for performing that analysis, and even the cost. Diba? So lagi kong sinasabi sa inyo yon. So if it is time efficient, therefore it can also be, or most of the time it is also cost efficient. Okay? And the availability of the resources or the inputs that we're going to use has something to do with the cost also. So it always affect the efficiency or cost efficiency. Okay? And then we have here letter B. So if necessary, develop new procedure. So yun yung ano natin, that is actually the focus of our our subject. No, because if there are procedures that is not or does not cater the things that you want for the analysis or for the result itself, you have to develop new procedure. Itong developing a new procedure, hindi naman ito basta-basta na gagawa ka ng bagong procedure without basing it with the original procedure or certain references that has undergone the same analysis that you are going to perform. So dito sa developing new procedure, you have to take note also that um, kaya nga sa mga researches or experiments later on, if you're going to be enrolled in research and you're going to pursue with experimental design, research design, you have to, or at certain cases, most of the time, you have to perform preliminary tests, no? Just to make sure that the procedure that you are following or the procedure that you are following or the result that you want to achieve is achievable or ano bang tawag, feasible for that sense. Okay? So if not, then you have to develop a new procedure based from your experience with the use of that procedure that you have chosen, or you can develop a new procedure. What do you call this? You have to develop a new procedure that is based from other references. Okay? Laging ganon sa sa pag-develop ng new procedure. Okay, if necessary lang naman. However, if the procedure actually fits the test that you're going to perform, then so be it. Diba? Huwag na tayong magbakahirap kasi meron ng established procedure for that matter. For example, test for carbohydrates, test for proteins, mga ganon. So test for lipids and among others. Okay? There are actually established procedures for those. No? What else? So for step 3, that is sampling. So sa statistics or biostatistics nyo later on, you'll be discussing different techniques on sampling. Merong random sampling, mga ganon. Tawag nito. Meron din yung random sampling na assigning of samples. Kasi yun ang yun ang point of interest or subjects, no, yung mga respondents for that matter. Yan, we have to select representative material to analyze. We should not use the entire sample. Bakit kaya hindi natin dapat gamitin lahat ng sample? Anong Hi, naririnig niyo pa ako?
Yes, sir.
Okay, ayan. So, I'm so sorry for the internet connectivity issue. We just have to be patient about it. Kasi hindi siya rare na problem when it comes to online learning. Anyways, you will be having actually your um, face-to-face already. I think this mid-year or first semester, next school year. Ayan, so we will be conducting an orientation on February 9. So February 9 for MedTech, that is 9 to 11 a.m. Na-share na ba sa inyo yon with regards to face-to-face? Hindi pa na-share sa inyo?
Sir, yung ano po?
Yung face-to-face.
Sir, wala pa.
Sige, babalita siguro sa inyo yan. But we are actually creating, we will be having an orientation for parents and students on February 9, both for MedTech and Nursing and even BS Bio for SHANS, limited face-to-face classes. So all of the students who are vaccinated can attend or can go to the school for limited face-to-face classes. So for ano lang naman to, for hindi naman lahat ng subjects, but for subjects that needed laboratory skills, laboratory, what do you call this? Building up these skills you need for your future career. Hindi naman lahat. So, yung mga ano lang, skill-related subjects. Okay? Ayan, sorry for the interruption once again. At least, am I audible on your part now?
Yes.
Yes, sir.
Okay. Yeah. So, let's continue our discussion. I'm so sorry for that. Ayan. So, going back to my question, what do you think are the consequences if we use the entire sample? Oh, do you have any idea on what will be the consequences if we use the entire sample? Pag ginamit natin lahat ng sample na na-collect natin, for example, ano kayang consequences? Ano yung magiging effect? For example, nagkamali ka. Okay? Oh, ano kayang consequences nun? Yes, Lizel.
Sir, hindi na po siya pwedeng ma-duplicate.
Yun. So, di na siya pwedeng ma-duplicate. That's why, when performing analysis or okay lang, analysis with regards to your experimentation or yan, mga different when you try to perform tests later on, you have to make sure that you still have the sample, the prepared sample. Okay? Hindi dapat nyo ginagamit lahat yon for just only one trial or experiment or analysis. Because once you have committed mistake or error, it might be affecting the reliability and even validity of your test result. That's why it is recommended that you can either duplicate the trial or even triplicate it. Okay? Yon. Duplicate or triplicate your analysis. Okay? Kung magkaiba man, hindi kasi always, all the time na pare-pareho yung nakukuhang result. Bakit hindi pare-pareho? Of course, there are certain factors that may affect a result or a test. So, ang gagawin natin, no, yung mga factors na yun, we have to minimize all of them para maging 99.99999%. Remember, as just like what I have told you, science doesn't have 100% assurance. There will always have a percentage error there or margin of error as they term it in statistics and so on and so forth. Okay? Yon. It is advised that we have to duplicate or triplicate our trials so that for that sense, for example, yung una natin, we were able to measure 15 grams. Yung pangalawa, 14.9 grams. Yung pangatlo, 15.1 grams. So, anong gagawin natin doon? Remember, tignan nyo yung nakuha nating measurements, magkakaiba. So, what are we going to do with those three collected data? O anong gagawin natin doon? Paano natin nare-report yun magkakaiba? Yung nakuhang measurements. Yung unang trial, 15 grams. Yung pangalawa, 14.9. Yung pangatlo, 15.1. How are we going to report? Yes, Zanya?
So, hindi ko sure, pero yung average ba? Yung
Yeah. Yes, so that is correct actually. So you are sure with your answer. You just have to be confident, okay? Ayan. So, yon. We have to get the average or the mean. For example, yung mean, lagi naman pag experimental mean ang kinukuha. For descriptive type of research, for that sense, pwede rin yung mode ang gamitin or even the median. Okay? So, ganon. Ayan, we have to duplicate and triplicate our trials. Okay? Ayan. So, consistency in sample collection. Yan, we always have to remember the protocols in collecting the sample, preparing or collecting the sample, transporting the sample, and even what do you call this? Preparing the sample. No, that's our next step actually. Step 4, sample preparation. So how do we convert our sample into a suitable what do you call this? Suitable uh, um, component or suitable what do you call this? Compound necessary for chemical analysis. Okay? So we have to convert this sample. Okay? So sa large population, kumuha tayo ng, for example, sa population ng 10, kumuha tayo ng 5. Yung 5 na yon, i-convert natin into a, for example, how do we convert this sample preparation? Okay? Dun sa 10 liters ng body fluid na yon, we only get 5, for example, and then we use, for example, mga 100 microliters, 200 microliter, mga ganun na blood. Okay, before we do that, we have to convert the body fluid into a suitable form, okay, for chemical analysis. So how do we do that? Yung mga na-present kanina like extraction, precipitation, and among others. Okay? So there are a lot of preparing sample preparation that we're going to discuss later on. But these are the most common. Extraction actually ang pinaka-common. Extraction and um, precipitation. For precipitation purposes, ito yung mga ginagamit, for example, sa metal analysis, um, what else? Tawag nito, even sa urinalysis perhaps, yun, sa detection of proteins, detection of blood, and even detection of carbohydrates in urine, and even fecal analysis sa feces. No? Most of the time, precipitation ang ginagamit doon for sample preparation. Okay? So, of course, we have to convert also the sample into suitable form like uh, aqueous solution. Okay? And even we have to concentrate the sample. Yung concentrate sample, yun yung after the extraction process, that is actually a concentrated sample. Okay? Or ginagamitan din siya for sample preparations. We also use the basic techniques like filtration for that matter, decantation. So makikita nyo dito sa figure natin dito, we have a lot of basic techniques that we use. So that's actually a collection of basic techniques. Decantation, centrifugation, um, what else? Filtration, for example. So those are necessary techniques needed for sample preparation. That's why it's good that I discuss them in your inorganic and organic chemistry. Okay? So these are just sample. Yung pangatlo natin, sorry, third, ito ha. So yung pangatlo natin dito, remove species that interfere with analysis. So we always have to convert the sample into a very specific, for specific chemical analysis. Although meron din tayong mga type of tests that are selective and yung na-discuss natin kanina umaga, selective and specific, no? We can skip this one. However, most of the time we have to make sure that the constituents that are present in the sample doesn't interfere with the analysis. If that's so, then you have to remove those species by, ang ginawa niya dito ay centrifugation. Kung titignan nyo, ayan, he tried or she tried to mix the chocolate with a solvent, no, organic solvent, then mix and then centrifuge, tas i-decantate niya, yun. So maiiwan yung residue, okay? Yung liquid ay, for example, yung liquid, yun. Ang kailangan lang natin is yung residue. Then we have to remove or itapon yung um, liquid na hindi naman kailangan for analysis. No? That will save time and of course resources, and even yan, it will save time and resources. Okay. Dito naman it talks more of experimental setup like measuring the tumor size by fluorescence technique. Okay. Ayan. Although it's just one example of technique, no, they used to what do you call this, pinpoint or measure the tumor size by using quantum dots. Okay. However, there are also other techniques that are used um, specifically for the infusion of drug dosage or extracts in this laboratory animals, no, performing scratch tests for ointments, etc., etc., and even tawag doon, and even yung paggamit natin ng mga what do you call this, modified organisms, or yeah, mga genetically modified organisms. Very ano na yung mga yon, very common na yung mga yon kasi we tawag nito, with the different processes involved in biotechnology, the advancement in biotechnology, mga procedures doon, okay? Um, madali na nating what do you call this? Madali nating makita, ma-measure, makita yung effect kasi meron ding mga transparent na mga lab animals na ginagamit, no? Like the sa mga for phytochemical screening or sa toxicity, ganyan, yung mga brine shrimp, zebrafish, and among others. So those are the most common lab animals they use for these processes. Okay? And aside from those, tawag nito, of course, with the use of animals along the process, we have to consider the ethical consideration. So yung the ethical way. So if you remember during the siguro mga 80s or 1700s, yung timeline na yon, um, that's the first, although hindi yun experimental na yung ganito, like the infusion of drugs, ganun, that's more of psychological observation for that matter. Yung gumamit sila ng infants or baby. Okay? So they use that and then it is subjected into criticisms, ganyan. So we have always to consider the ethical way of performing tests, not only to animals, but of course, not only to humans, but of course to animals. No, because life is precious. And recently, if you heard the news about um, using human as a test organism for COVID, if you heard the news on exposing the person, the test organism to COVID, and then observe, no, the mechanisms that they will be going through, mga ganon. So they actually start doing that. Ngayong ano lang ngayon, ngayong year lang 'yon. Okay? So ano bang mga questions that we need to answer in preparing samples? Okay? So anong mga questions dito? These are general questions like, is the drug active? Does it cure the disease or illness? How is the drug taken? Is it by pill or injection, by IV? So how often does the drug need to be taken? Does the drug have side effects? So these are the things that we want to know first before we proceed to experiment or data gathering. Okay? So how these questions are typically addressed? Ayan. Oh, ethical consideration. Treat animal with drug. Okay, we have to not only infuse na saksak tayo ng saksak or inject tayo ng inject. Okay? We also have to monitor the drug duration, the location of the drug accumulation, and even, of course, most importantly, animal health. Diba? Our health. Okay? Yun. So that's why we always have to consider the ethical way of performing tests. Okay? Most specifically for humans. Okay? And yon, that's a very rare case that they use a human as a test organism because it is it is tawag nito debatable actually. So what if it compromises the health of the test organism, then you have to be responsible of that. Okay, so remember, once na compromise ang life, we cannot be able to return it into the way it should be. 'Di ba? So kung nag-complicate na, for example, ang test organism ay human, then then we cannot do anything to revive, for example, kung very complicated yung case. Okay, that's why we have to monitor especially the health of the test organism. No, not only yung purpose natin like the location of the drug, if the drug is able to treat like this. Okay, kasi most of the time yung mga rat na for example, yung mga animals like rat, mice, rabbit, etc. They are being actually introduced to the disease, no? For example, yung rat naman ay healthy. Tapos we are going to test whether the blood sugar of this increases when we infuse like this or like that. Ang una namang ginagawa doon is we induce them into that kind of situation. And then i-treat natin. Ganon. So, ganon naman always. Okay. During my college, we were able to use lab rat, lab mice, or lab rat, even rabbits and ano yung isa? Yung mga maliliit, parang rabbit din yun eh. Pero wala maiksi yung buntot nila. Tsaka cuties yung mga yon. Cute size. Ano yung tawag doon?
Hamster po.
Parang hamster ata yun. Meron pa yung isa.
Guinea pig po.
Yon. So, guinea pig. So, yon yung mga nagamit namin when I was in college. No? So, ganon. We have to treat them the way we treat ourselves. Okay? So, how do you treat the animal with the drug? How do you monitor the drug concentration in the animal? So, how do you determine the drug location? And how do you determine the animal's health? So, all of this is subjected into preparation. Okay, we have to prepare everything before we conduct the experiment. So dito naman, these are just um, tawag nito, sample processes they do for preparing samples for drug discovery. See if the drug is able to treat the illness or the disease, perhaps. Ayan, so they do chromatography, what else? Cross section. Although ngayon, even if we do not tawag nito, if we do not complicate the life of the organism, we can able to detect because of the advancement of technology that we have. Yung mga scanning devices natin is, I think, advanced. Even in the Philippines, I think, in um, the NCR and the northern part, and even sa southern, meron na tayong mga advanced tawag nito, scanning devices. Although, of course, if we compare Philippines with other countries, then perhaps the countries like US and other European countries and sa North America and South America, of course, no, we are far beyond them. Okay, but of course we are doing our best to be globally competitive naman, 'di ba? That's why we have sa curriculum, we have to develop students in terms of their research skills, no, being a globally competitive profession someday. Ayan. So that's why kung maalala niyo yung kanta na ano yon, yung nag-a-abroad sila kung na 90 kanta. Anong title nun? Kung maalala niyo kung anong kantang yon, yung mga professions, no? It talks about professions going abroad. Ganon. So, yun. Yun yung mga kailangan nating um, mag-reflect, no, na mga bagay. Although, mataas nga ang compensation sa other countries, no? Of course, we always have to help our country, no, in terms of health, education, and among others. Yan. And the step 5 is the analysis or the experiment process, experimental process or the analytical process, the analysis, performing analysis. Okay? So, dito we have to replicate measurements. So, yun yung very ano dito evident, no, when it comes to performing analysis, we have to replicate our trials to avoid large errors and even the reliability of our results. And take note, no, in preparing, sorry, in preparing the samples, we have to subject everything that we use, like the equipment and even the reagents, into calibration, the standard way of calibrating the equipment and even the reagents. So we have to use the standard always. And with that, we have to um, tawag nito, compare the result that we get from a standard result. So ano yung mga indications? For example, there is a change in color from yellow to blue. So this indicates that limonella or oxalate is present in the compound. No, those are indications that we need to um, base with to report our results. Okay, ayan. So report and interpretation of data. And then after we do the report and interpretation, of course, we have to remember, sabi ko sa inyo kanina, that in dealing with reports and interpretation, we have to consider the understandability or how our clients understand the results or the interpreted data that we have gathered. So hindi basta-basta ibibigay natin yung raw data, di ba? So there are many ways on expressing or reporting a result, either by tabular way or graphical way or even the qualitative descriptions. That is why after the laboratory test, yung result ibibigay sa attending physician or even the nurse, oftentimes. And then yung test result na yon, it is being explained, okay, very well in the common language, okay, that is understandable at the client's end. Okay? And then of course yan, how the report is used, how do we draw conclusions? Okay? That supports the tawag nito, that supports the um or that answers the problem or the question. Okay? So anong concentration nito? Kaya, 'di ba, balik tayo dun sa problem natin. So anong concentration ng arsenic sa water? For example, 42% by mass in water during the water analysis, no? The concentration of arsenic is 45% by mass. So it indicates that like this, like this. Okay? So ganun ang conclusion. So therefore, if there is this kind of percentage in water, the water is not potable. Okay? Meaning to say this, it cannot be hindi siya naiinom. Okay, so how do you treat that? So yun naman. Kaya nga sabi ko sa inyo, if yung general problem natin, merong mga specific questions. So what is the concentration of, after determining the concentration of arsenic in water, then paano ngayon i-convert yung tubig na yon into a potable one, drinkable. Okay. So how do you eliminate the arsenic element in that? So there's comes another problem. So you have to propose another solution to eliminate or remove arsenic, or even how do you introduce an intervention? Meaning to say how, for example, magbigay ka nung anong binibigay nila ngayon pag nagkakaroon ng flooding. Meron yung kit na yon, na yung kit na yon ay parang small filter na kahit marumi yung tubig, tapos pag na-filter, pati mga microorganisms na filter. So the remaining one is a potable fresh water. So meron na ngayon na mga advanced technologies like that. So ikaod mo lang ay hindi dirty water, for that matter, yung mga flood water, no, after ng sakuna, tas antayin mo lang yon. You just let it sit for quite some time, like 10 minutes or 15 minutes. Then you can have a potable drinking water, fresh water. So may mga ganong ano na ngayon, technologies. But of course, those are sabi ko nga sa inyo kanina, when it comes to quality, okay, you get what you paid for. Okay? Yun. So these are uh, just some of the roles of analytical chemistry. Ayan, it's not only used in science and technology to solve practical problems, but of course it is applied in all areas of sciences and industry and even medicine. So for example, ayan, so determination of concentrations of oxygen and carbon dioxide in the blood samples. Ito, kung meron kayong mga sasakyan, yung mga nagpupunta sa emission testing center, no? That facility actually determines the quantities of hydrocarbons emitted by your automobile, for that matter. Okay? And then that is subjected into different tests. So pag ganito, high level of quantities ng hydrocarbons that is emitted by the exhaust gases for devices, then you have to calibrate the tawag nito, you have to calibrate the machine, yung makina ng sasakyan. So ganon. So dapat makapasa siya sa emission testing center, emission testing. Okay? And then quantitative measurements of ionized calcium in blood serum help diagnose, yan, mga more of determining the metal analysis. Okay, mga trace elements or major what do you call this, major elements or trace elements that are present in our blood serum. Ayan. And even determination of the amounts of nitrogen in foods. Okay, we are talking here about nutritional facts or nutritional value. So we have to look always or all the time to the nutritional facts or nutritional value of the food that we buy. Okay, and even the drugs. So dito naman it talks more of material science o material science sa engineering, um, the quality of the steel or the quality of the metals that you're going to use for construction purposes, mga ganyan. The strength, hardness, corrosion resistance, and even ductility. All of which, no, the characteristics, the desired characteristic you want for a metal for construction purposes, di ba? So the mercaptan content of household gases, yan, sa mga household materials natin, and even agricultural products like our fertilizers, what do you call this, and other chemicals needed for the development and growth of the plants that we culture. Yan, lalong-lalo na ang visca is one of the although it's not the rice bowl of the region or the Philippines, but tawag nito, agriculture is very provident here. Very, very evident here in Nueva Vizcaya, di ba? So, yon, lahat yon, no, are different roles of analytical chemistry. So, in our lab code later on, I will also be posting a uh, 3 videos pertaining to how analytical chemistry is important not only in environmental awareness and or environmental control, in industry and even pollution control, but of course in health, okay? Healthcare. O, ano pa? That's why ang analytical chemistry is considered to be as the central science because all subdisciplines, not only in science, no, rely on analytical chemistry to function. So kung makita nyo dito, there are certain fields here that are subdisciplines that need, okay, that need analytical chemistry. Okay, yan. Meron tayong geology, environmental science, agriculture, social sciences, material sciences, medicine, engineering, physics, chemistry, and even biology. So sort of a various field of discipline. Okay? So, ayan. That's why analytical chemistry is interdisciplinary in nature, no? Because of the chemical analysis, which makes it a vital tool or vital important um, tawag nito endeavor, no, not only in medical, industrial, government, even in the academic laboratories throughout the world. So yung academic laboratories, it can be the laboratories that are present in the school. Okay. So those are the things that we can learn from this presentation. So do you have any questions before we end our session for this afternoon? Meron ba tayong mga question? None so far? Nakatulog naman na ata kayo. Wala namang questions? Can I have a heart or can I have a response?
None, sir.
Ayan, okay. Ayan. So, if you do not have any question with regards to our discussion and your quiz for Monday, then I guess we can call it a day. No, because I will be attending a defense. So, yung mga nagde-defense ngayon is yung mga ate at kuya nyo, mga third year. So, I hope to see you later on sa proposal defense also. Okay? So, thank you so much for your participation and do not forget to answer our attendance. So, I will be posting also our attendance in our LMS. And then, I will also be posting the reflection paper, the different questions. Essay format yun ha. Yan. So, don't forget. That will be submitted on February 11. Okay, so with that, thank you so much and I shall see you next week. Goodbye, everyone.
Goodbye, sir. Thank you
Bye
po.
po.
Bye.
Sir, thank
Thank
you.
you, sir.
Okay.